All right, so our last new topic in these study sessions is, first of all, talking about how embryos develop. That word up there in the beginning there, you may not recognize, metazoan. Here we're talking about animal cells, or animals, that are multicellular and eukaryotic. That's essentially what metazoan means. So it's animals, multicellular, eukaryotic. What we're mostly going to be looking at today is how an embryo forms and how it develops from that point on. And it starts out as a 4-step process. Step 1 is fertilization. And we know that in fertilization you have a haploid sperm getting together with a haploid egg to make what? What kind of zygote? Diploid, okay? Haploid sperm, haploid egg, getting together to make a diploid zygote. And that egg has been dormant, until it gets fertilized. Once it gets fertilized, it starts dividing really, really, really fast. And we'll give that a name in a minute. It also starts to divide and make 3 axes, 3 directional points inside the embryo. And those are anterior and posterior, dorsal and ventral, and left and right. What's the anterior? Think of a 4-legged animal, like a dog. What's anterior on a 4-legged animal? The front, like the head side. Posterior is like the tail side. So front to back is anterior, posterior. What's ventral? Somebody tell me what ventral might be. Where's ventral? Yeah, that's like belly side, okay? And then, so ventral to dorsal. Dorsal is like along your backbone. So belly to back. Dorsal, ventral. And then left and right. So step 1 of this process of embryonic development, fertilization, haploid sperm getting together with haploid egg to make a diploid zygote. The cell starts splitting really fast, going through rapid cell division, and it's mitosis that it's going through. And it also establishes these 3 axes. Step 2 of the process, we number them, 1. Step 2 is called cleavage. What does cleave actually mean? What does that word mean? Not quite. It can mean that. But typically the word cleave means to cut, to break, to tear. In this case, it means to split. So what we said is that fertilized egg, the zygote, it divides so fast in this cleavage stage that there's no time for the cells to grow. So even though it's making a lot of new cells, the zygote doesn't get any bigger. It's just dividing itself into small pieces. So the cleavage is rapid cell division, where the zygote divides itself into smaller and smaller cells, but there's no net growth of the entire zygote. One of the first things the zygote does is it organizes itself into this ball of cells, a solid ball of cells called the morula. And it's that ball of cells is usually a 16-cell structure. So that's one of the first organizational things that it does. The cells organize themselves into this solid ball called the morula, a 16-cell ball. Then those cells start making a fluid that concentrates in the middle of the ball. And it pushes all the cells from inside to outside. So essentially the ball hollows itself out. It's got this fluid core, and then the cells on the outside. Once the ball hollows itself out, it gets a different name. It's called the blastula at that point. So it starts out as a zygote, one cell, divides really fast, organizes itself into a 16-cell solid ball, that's the morula, and then those cells secrete fluids, which push the cells from the inside out. Now the cells are on the outside of the ball, and fluids on the inside. At that point, it's called the blastula. And that's pretty much where we're going to stop with cleavage, step 2. Step 3, the cells start organizing themselves into layers. Step 3 is called gastrulation. And what ends up happening, the cells organize themselves into 3 layers of cells, and they're called the germ layers. Don't confuse these with germ cells. What are germ cells? Cells that can do what process? Meiosis. This is a different kind of thing here. This is not meiosis related at all. These are the germ layers. These are essentially the layers of stem cells in the embryo. These are the cells that will make you. And each layer is in charge of making different things. So, again, the cells of the blastula move around and they migrate and they organize themselves into these 3 layers. We call the layers the germ cell layers. So there's a layer on the outside, a layer of cells on the outside. We call those cells the ectoderm. There's another layer underneath that. We call those cells the mesoderm. What's meso mean? Middle layer. And then there's an inner layer as well. And those cells on the inner layer we're going to call the endoderm. So those 3 layers of cells in the embryo are the germ layers. And those are essentially your stem cells. They're going to make you. And each layer is going to make different parts of it. Another thing that happens during this stage is that at one side, this embryo starts to pinch in on itself, starts to invaginate. And the opening into that ball is going to become what? What's the opening into that ball of cells become? In some things, that opening becomes the mouth. In some things, that opening becomes the other side. That's what happens with you first. The anus forms from that initial opening. Depends on the type of animal. We'll get to that in a minute. Notice, though, that opens into a cavity that looks an awful lot like what? What does that sort of look like? What do you think that opening leads to? What does your mouth lead to? What does your anus lead to? Your digestive system. So this cavity, sometimes it's called the arc enterine or primitive gut. It's the beginnings of what will become your digestive system. So the opening into the ball of cells, initial opening, I don't think I gave it a name. That initial opening is called the blastopore. That's the name of that opening, the blastopore. That opening in some things becomes the mouth, in other things the anus. And the cavity that it leads into, the arc enterine or primitive gut, eventually will develop into your digestive system, or at least part of it. At this point, the ball of cells, once it's organized itself into these 3 germ layers, has yet another new name. At this point, it's called the gastrula. So we've got 3 names for the ball of cells. Starts out as a morula, 16-cell solid ball. Then it hollows itself out. At that point, it's called the blastula. And then finally, the cells organize themselves into these three distinct germ layers. At that point in time, it's called a gastrule. So three different names for the ball of cells. And then the last stage of embryonic development that we're going to mention, step 4, is organogenesis. And that term literally means what? Organogenesis. What's Genesis mean? Creation. So creation of organs. Remember that each one of these germ cell layers is in charge of making specific organs and organ systems. And that's what's going on here. They start to make you. And one of the very first parts of you that's made is the neural tube. And what do you think that neural tube becomes? Neural tube, what do you think that would become? The brain and spinal cord, the central nervous system. That ought to come first, right? That ought to be one of the first things you make. So what you need to know about organogenesis is it happens after gastrulation. The three germ layers start to make the organs. They start to create the organs. And one of the very first organs or pieces of an organ that's made is the neural tube, which becomes your brain and your spinal cord. And that's about as far as we need to go with it for right now. Questions there? All right. Can I proceed? That's a picture of a blastula. And you can see that inside is hollowed out, all in the middle. There's cells on both sides, but hollow in the middle. Here's a gastrula. And remember, the gastrula has organized itself into three layers. You got the layer on the outside. That's the ectoderm. There's a layer on the inside, so that'd be like right in here. That's the endoderm. And there's a layer in the middle, and that's mesoderm. And those three germ layers are the parts of your body in charge of making you. Each layer makes different parts of different organ systems and such. Again, this hole here is the blastopore, and that leads into this cavity right in here. And that's called the archenterine, or the primitive gut, and that becomes your digestive system. All right, another term here we need to mention, two terms, protostomes and deuterostomes. What does the word stome mean, like in stomata? What is a stomata? The hole on the bottom of the leaf. The point is, it's an opening. Stoma can mean opening. Really, in this context, that means mouth. And what's proto mean? First mouth. So protostomes are animals where the blastopore becomes the mouth. The mouth forms first. And that includes a wide range of animals. I'm going to mark off a couple of these because they're technically not correct. So let's include just those as protostomes. Anybody know what annelids are? Annelida. What kinds of things are in a group called annelida? It's a phylum name. Any ideas what annelids might be? These are segmented worms, like what would be a segmented worm? Earthworms, leeches, stuff like that. What about mollusks? What are those? Somebody in here I know knows what mollusks are. What are mollusks? Clams, oysters, also snails, slugs, also octopus, squid. Mollusk. That actually means soft-bodied. Kind of gooey, slimy body. And the protostomes also include arthropods. Arthropods are mostly what? Most common arthropods. What does the word arthropod mean? Pod means P-O-D, what does that mean? What's full of answers? Pod means foot. Arthro means jointed. Jointed feet. These are insects. Also spiders. Also crustaceans like crabs and stuff. These are protostomes. In these three phyla, the blastopore becomes the mouth, and it forms first. We'll talk about some other distinctions between protostomes and deuterostomes in a minute. But for right now, protostomes, blastopore becomes the mouth, forms first, and it includes the segmented worms, phylum Annelida, the soft-bodied creatures, mollusca, things like clams, snails, slugs, oysters, and also arthropods, which are things with jointed appendages like insects, crustaceans. Deuterostomes, on the other hand, are what you are. What does deutero mean? Deutero means second. Where have you heard that root before? Deutero. Deuteronomy, the book of the Bible. Why does it mean second? Because it's not the second book of the Bible, right? What book of the Bible is Deuteronomy? Like Sunday school person tell me. Genesis, Exodus, Leviticus, Numbers, Deuteronomy. That's the fifth one. Why is it the second? Why does it mean second? Why is it given a name that means second? Maybe, but the main reason it's the second place in the Bible where you can find what? I know you're getting Sunday school listening. Where you can find the Ten Commandments, the law. The second place in the Bible where you can find the law. Originally up here in Exodus. Anyway, Deuteronomy is a group of animals that are an evolutionary line of coelomates. That's probably another new word for you. Anybody know what that is? A coelom. No. I don't know anything about animals, do you? A coelom is a body cavity. Body cavity. Place to put your organs. There are animals that don't have body cavities, largely because they don't have what? Organs. Things like sponges, for example. Jellyfish. They don't really have organs. But we do. We're coelomates. We have body cavities, places to put our organs. The only organisms that fall into this group of deuterostomes are echinoderms and chordates. You are a chordate. What does it mean to be a chordate? Vertebrates. Chordate, that's a little bit of an oversimplification. But chordates are essentially the vertebrates. So all the vertebrates, from fish to amphibians to reptiles to birds to mammals. Those are chordates. What about echinoderms? What are they? What's an echinoderm? The word means spiny skin, literally. Echinoderms are things like starfish and sea urchins, sea cucumbers, that kind of group. And what this means, because our embryos develop in such a similar way, that means other than the other vertebrates out there, according to biologists, your closest relatives are things like starfish and sea urchins. Take away the other vertebrates, your closest relatives are starfishes, sea urchins, and sea cucumbers. So let's talk about a couple of other differences here. But in deuterostomes, that blastopore, that opening, becomes the anus, and it forms first. That's one of the main differences between the two groups. Mouth first with protostomes, anal opening first with deuterostomes. So there's also a couple of other differences too in terms of how the embryos develop. Up top, let's talk about protostomes first. The protostomes, their top of cleavage is said to be determinate. They have what's called determinate cleavage. So very early on in their embryonic development pattern, what's already been determined about the cells? What do you think determinate means here? What's already been determined right after the cells divide initially? What they're going to be. What they're going to be when they grow up. And how does the fate of a cell get determined? Methylation of the DNA, right? DNA. So even by the time there's only 4 cells in this embryo, the fate of those cells has already been determined. Which means they're really no longer what kind of cells? Really no longer stem cells at that point. They've already been methylated. There's some things they can't do at that point. Remember, protostomes are things like arthropods and annelids and mollusks, not you. Deuterostomes, on the other hand, have indeterminate cleavage. I'll have to work on that. That's terrible. Indeterminate. What that means, early on, as the cells divide, no methylation happens early on. Which means those cells are all still what? They're all still stem cells. They can all still become whatever they need to be. If you were to take out one of these first 4 cells, throw it away, these 3 can still grow into a normal embryo. Up here, if you take out one of these first 4 cells, the thing dies. Because by this point in time, the fate has already been determined. So our cells have a little bit more leeway to become what they want to be. It's further in before they're methylated and their fate is determined. Also, the way the cells divide, the pattern they form new cells in is a little bit different. Protostomes have what's called spiral cleavage. Cells form sort of a spiral pattern as the embryo is growing. And on the other hand, deuterostomes have what's called radial cleavage. So 3 differences between deuterostomes and protostomes. Protostomes, the mouth forms from the blastopore, and it forms first. And it leads into the arc enteron, the primitive gut. Cleavage is determinate in protostomes. The fate of the cells is determined really, really early. They're really no longer stem cells at that point because a lot of their DNA has been methylated. And the cells cleave in a spiral pattern in protostomes. Whereas in deuterostomes, that initial opening into the embryo, into the blastula, the blastopore, becomes the anus. It opens up into the arc enteron or the primitive gut. Cleavage is indeterminate, meaning that the cells retain their ability to be stem cells for much longer. And the cells form in a more radial pattern rather than in a spiral pattern. Also remember that the protostomes are things like segmented worms, annelids, arthropods, insects, and mollusks, things like clams and slugs and stuff. Whereas the deuterostomes are all the vertebrates, chordates, and also the echinoderms like starfish. Oops. All right, another thing that we need to mention here is what the germ layers actually do. Remember, the germ layers are layers of cells in the embryo. And each layer of cells has a very specific thing that it does. The endoderm is the innermost part of that gastrula, that ball of cells. So it would make sense that the endoderm would make parts of you that are very deep within your body, like internal parts. So the endoderm makes your digestive system. That's where your digestive system comes from. The endoderm forms most of your lungs, your liver, and also the pancreas. Parts of your body that are very internal, mostly. On the other hand, the mesoderm is the middle germ layer. It makes a lot of things that are sort of in the middle of your body. For example, the dermis. What's the dermis? It's the middle layer of the skin. What's above the dermis? Epidermis. And what's below the dermis? Called the hypodermis. There's 3 layers of skin. This is the middle one, made by the middle germ layer. Mesoderm also makes your bones, your cartilage, makes your blood vessels and your kidneys. It makes your reproductive organs, gonads. Mesoderm also makes the muscles and the linings of the body cavities. Most of your body cavities are lined with almost like a membrane. This is the structure that makes that. And then the final one is the ectoderm. And it's the germ cell layer on the outside. And it makes stuff mostly on the outer part of your body, like the epidermis, the top layer of the skin, the nervous system. It makes a lot of your sense organs, like your ears and your eyes. It makes the tooth enamel, the outer part of the teeth. And it makes the epithelium, which is like the lining of the mouth, the nose, the salivary glands, and the anal cavity, the anus. So again, ectoderm, outermost germ layer, makes things usually toward the outside part of your body, the outer layer of the skin, a lot of the sensory organs that are on the outside, like ears and eyes, the outer part of your teeth, the enamel, and the linings, the epitheliums of the openings into your body, like the mouth and the nose and the anus. So again, the germ cell layers are the layers of cells in the embryo that make you. And each one of these layers has specific parts that it's in charge of making. Make sense? Right. Give you guys a minute to finish up with this, and then we will move further.
How did they figure out which layers made what?
You know, I don't know the answer to that question. That's to some point they can grow these things in like a tube on culture, but not very far, not far enough to see organs. So my guess is they probably started out with something that's developing in an egg, like a hard-shelled egg, and watched it from there. But don't quote me on that because I'm not 100% sure. All right, so our germ cell layers are essentially our stem cells. Keep that in mind. Your germ cell layers are the cells that make you. That's what stem cells are. Remember that stem cells are cells that haven't differentiated yet. They haven't been methylated. They can become anything they want to be. By the way, what's a methyl group made out of? What does methyl mean? Chemistry people, what's a methyl group? It's 1 carbon connected up to 3 hydrogens, CH3. Methyl groups get connected directly to what? The cytosines in the DNA. And when they get connected, what does that cause the DNA in that area to do? What does DNA physically do when it's methylated? It clumps up, which means what can't happen to it if it's clumped up? It can't be transcribed, which essentially means it's off, right? Because if you can't make RNA from it, you can't make proteins from it. Essentially, it's off. So, again, your stem cells are essentially your germ cell layers. They haven't been methylated. They retain the ability to become anything they want to be. There's a lot of research going into stem cells. And for a long time, it was controversial. Why was stem cell research controversial for a long time? A lot of the stem cells they were researching, they got from aborted fetuses. And a lot of people were really, really uncomfortable with that. Because they were saying, we need these stem cells to do this research, so we're encouraging people to have abortions so that we can get them. And that bothered a lot of people. There are other ways to get stem cells, they are finding. They don't have to get them from aborted fetuses. So that's becoming less and less controversial over time because they're finding other ways to get the stem cells. But there's hope that these stem cells might one day be used to treat all kinds of problems. They've already been used to treat leukemia successfully. What's leukemia a problem with? Ultimately, your bone marrow. And what's the result of that? What does leuko actually mean? White blood cells. You have too many white blood cells and not enough red ones. One of the places that they've gotten stem cells from to treat leukemia is from cord blood. When I say cord blood, what cord am I talking about? The umbilical cord. When a new baby is born, that umbilical cord has lots of stem cells in it. And there's actually a whole industry that's sprung up lately where the doctors who deliver the baby can collect this cord blood. They send it off to a company that stores it for you. You have to pay them like a monthly fee, and they store this cord blood for you. And if you ever need it, they ship it to the doctor and they can use it. There's been several cases where like a sibling or something has gotten leukemia, and this cord blood has been injected into the bone marrow, and it's corrected the issue. The stem cells have differentiated into normal bone marrow cells and fixed the problem, essentially. There's hopes that it can be used to treat other kinds of cancers and all kinds of tissue damage. You guys don't know who Christopher Reeve was? Who was he? Yeah, he was the original Superman, the actor that played Superman. Not original. I mean, Superman's been around for 100 years, but in the first famous Superman movies, he was the actor. Kind of ironic that he ended up breaking his neck or his spinal cord. I'm not sure how. He fell off a horse, and he became paralyzed from the head down. And his wife, and he even, sort of became advocates, I guess is a good word to say here, for stem cell research, because they were hoping that they would be able to use stem cells to fix his spinal cord damage. He died a few years back, so that didn't happen. There's also some hopes that they can fix things like Parkinson's disease, maybe even things like Alzheimer's disease using stem cells. Essentially, put these cells into places that have been damaged or have issues, and then the hope is that these cells will grow back to be normal cells, and they'll differentiate into the cells that ought to be there. So that's what stem cells are, and they come from your germ cell layers. All right, we'll do this, and then we'll call it a day from this point. We mentioned body cavities a minute ago. And we said that the word for a body cavity is a coelom. There are a few kinds of animals that are acoelomate. And that simply means they don't have a body cavity. They don't need it, as I said a minute ago, because they don't have organs. These are things like sponges, jellyfish, maybe even some kinds of worms that we call flatworms. Don't really have very many organs, don't really need a body cavity. Then there are some animals, one group of animals in particular, one phylum, that has this thing called a pseudocoelom, which literally means what? False or fake. It's not really a fake body cavity, it's a body cavity, but it's made differently than the body cavities in all the other animals. This particular kind of body cavity, pseudocoelom, is made between two germ layers. It's made between the endoderm and the mesoderm. And that's different than the way it's made in other animals. We'll talk about that. This kind of body cavity is only found in a phylum of animals called nematodes. I know what nematodes are. Nematodes are roundworms. They're the most common multicellular things on Earth, and you may not have ever seen one. In a shovel full of dirt, just go out and dig up dirt. There's millions of them in just one shovel full of dirt. Little microscopic worms, usually microscopic, not always. You've heard of some of them before, though. You ever heard of hookworms? How do you get hookworms? How did you get an infection of hookworms? Walking around barefooted. And these worms burrow in through the soles of your feet and essentially become vampires in your body. It used to be really common in the South because people in the South couldn't afford shoes. Really, really common infection a long time ago. Not so much anymore. You probably heard of pinworms. Pinworms get transmitted a lot in daycares. And it's when a kid has pinworms, and then the worms make them itch around the anal opening, so they scratch. Well, we're talking about little babies. They probably don't wash their hands after they scratch. So then they go play with the toys. And what do little kids tend to do with toys? Put in their mouth. There's all kinds of worm eggs on that now. So they essentially ingest the worm eggs and they get pinworms, and it just goes around the daycare. Those are nematodes. They have, again, a pseudocoelom. It's not really a fake body cavity, but it's a body cavity made differently than body cavities are typically made. The main kind of body cavity, the regular coelom, found in most animals, segmented worms, mollusks, arthropods, echinoderms, and chordates. What makes it different is it's lined with cells that came from the mesoderm. So the inner lining of this body cavity is actually tissue from the middle germ layer. That's the main kind of body cavity, holds your organs, and again, it's in almost all the multicellular animals: annelids, mollusks, arthropods, echinoderms, and chordates. I'll come back to this slide in a second. I want to mention one other word before we go. I'll let you get this first. The one other word I want to mention is this one right here, cephalization. Whenever you see those letters, C-E-P-H-A-L, anybody know what that refers to? Cephalo. Cephal. It means head. This word cephalization means that animals typically have sense organs that are mostly concentrated in their head. It's the concentration of sensory organs into the head. Most of your sense organs are there. And especially if you're a four-legged kind of animal or you're crawling, it's pretty important that your sense organs be in the front. Otherwise, you may run into trouble. So it's the concentration of sense organs in the head. It's kind of protective because it lets you sense what's coming in front of you, and hopefully your head is in the front. And that is where we'll call it a day for today. Make sure I get the sign-in sheet, whoever has that, to get this recording stopped first.